In the past, biological research believed that life substances such as proteins store information, but soluble proteins were not truly regarded as having language functions. Taking soluble polypeptides, proteins between cells as cell language and analyzing and researching them by applying linguistic principles, what discoveries will there be? As a language, it should have a grammatical structure and perhaps functional structural components similar to punctuation marks. Could it be the site of protein glycosylation? Purposefully looking for these functional components is crucial. It can divide a long protein sequence into smaller fragments and eventually become functional units, facilitating in-depth research, deciphering, and understanding of cell language. The segmented cell language is similar to paragraph sentences or programming languages in computer programming. With more and more protein amino acid sequences being analyzed, using a computer to compare the relationship between amino acid sequences and functions can find common functional parts. The advent of AlphaFold focuses on the 3D structure of cell language and proteins, and at the same time pays attention to the spelling characteristics of language, which will help gradually decipher cell language. To further explore the intracellular process of cell language, it is found that cell language has astonishing similarities with the text processing of human language. The half-life of RNA is very short and it is extremely easy to degrade in vitro. Why is this? In fact, RNA is a random storage device for chemical or physical information processes in cells and disappears as information activities in cells stop. The DNA library constructed by conventional molecular biology is only cloning some transient mRNA fragments. The 23 pairs of chromosomes in human cells have different lengths. Why is this? We believe that the chromosomes in the cell nucleus are the hard disks for information storage in cells. The nucleus is spherical. Where the cross-sectional area is large, the diameter of the hard disk is large and the chromosome is long. Conversely, where the cross-sectional area is small, the diameter of the hard disk is small and the chromosome is short. At the beginning of individual development, genetic information solidified in specific base sequences has been stored in chromosome hardware. A large number of repetitive sequences form the remaining space of the chromosome hard disk, so that physical and chemical information is stored on the chromosome hard disk to form the cell's autonomous memory. This analogy not only has theoretical significance but also practical application value. For example, the transgenic method of transferring exogenous genes into cells is like importing information from a computer floppy disk into a hard disk. Applying the floppy disk or USB input mechanism in computer theory to transgenic practice may correct the blindness in the current gene therapy process, increase the efficiency and controllability of gene transfer, and make gene therapy widely promoted and applied in the end.